Longitudinal imaging is used in preclinical studies to follow the progress of a disease or measure the effect of a therapeutic. In oncology, optical imaging methods provide rigorous tools to monitor tumor growth and deliver precise quantitation of cell growth or gene expression at each time point in such a study. However, these optical techniques cannot directly account for changes in the underlying structure. These anatomical changes can be measured using high-resolution techniques such as micro-CT. For longitudinal imaging, a low X-ray dose is essential to minimize the effects of the cumulative radiation on the underlying biology. These optical and micro-CT images can then be co-registered to provide a combination of functional and anatomical data, ensuring that the maximum amount of information is extracted from the animal model.
Today we will demonstrate how to monitor tumor metastases and follow osteolytic lesion development using optical imaging and micro-CT. The Quantum FX performs CT at a very low radiation dose. This allows for longitudinal monitoring of osteolytic lesion development with a cumulative radiation dose far below LD50 levels.
In this experiment, cells from a human mammary tumor cell line expressing the luciferase gene will be used as an optical indicator of tumorigenesis in vivo. This cell line was created from a spontaneous lymph node metastasis that originated from a D3H1 mammary fat pad tumor and is known to aggressively form metastases. The cells readily grow in standard media with no need for selection of markers. Cells for intracardiac injection are prepared and luciferase activity is verified by standard methods as shown previously. Prior to intracardiac injection,